out. So, this is a talk I've given a few times, and it's on spatial biology, essentially, and a class of problems that I've gotten really interested in now. And I'll tell you more about it, the context and the mechanics of it. No, and part of this talk is also a situation of maybe biostatistics, more context dependent with AI and how that can play together, and sort of my take on things. It's a way, not the right way of things. So. No. With that said, I actually have a QR code here if you want to follow along, and then that'll link up to the slides as well. And given the link to Alison, and she'll be sharing this as well. So just so I make sure, can folks on Zoom hear me? No, if you can't hear me, then please unmute yourself and let me know. All right. So. Have this working. You can click on it. There you go. So I'll start with a very high level view, so that I want to get to the why of it before I get to the how of it. In spatial modeling, in these biological systems. So I mean, a lot of you might know here better than me that biology happens at different scales. I think what we see at the organism level is the sort of the tissue level, right? And then when you trace it back, a lot of action actually happens at the cellular or even the subcellular level. So to give you an idea, I'll use a mouse here, so the folks can zoom, can also follow. So to give you an idea of these, cellular and subcellular, we are talking actually microns here. So we are really zooming in at basically the interior of the cells and how these things. So actually, I read this book. I started off this book reading it as for fun, and this is by Siddharth Mukherjee. This is the third book he's written. The first one being Emperor of All Maladies, which is the history of cancer, and it actually ended up being a really nice sort of contextualizing a work. A lot of the work, that as I was getting into this field. Cellular therapies and what the future is and how that's evolved as well over the years. So I work in cancer. It's a disease system I worked in almost 20 years, and there is an increasing recognition in cancer of what is called the tumor microenvironment. And what that essentially means is actually an ecosystem of cells. A tumor is a living, breathing collection of it's a mass in the body. And then it consists of many different types of cells, and each cell has its own function. That's the T cell, B cells. Even in non-cancer, they have specific functions. Your body fights against it, especially in this context of immune reactions and such. So there's a lot of different types of cells, and the key point here is there's a constant interaction with them. So what the interaction means that, positive interaction would be them coming together, and then negative would be sort of repulsing each other. So in cancer, there's the cancer happens, tumor cells form, and then the hypothesis is there's a systematic recruitment of these different types of cells, and some are good, some are bad, right? And I have a link here. I won't go into it, but if you can see, there's something called a natural killer cells. So if you activate a natural killer cells, it can actually kill a tumor cell. So you can actually enhance. And why that's relevant is, especially in immunotherapy, which is sort of a new paradigm. It's not new anymore, but over the last 5-10 years, and how does immunotherapy? One, it works in certain cancers. It doesn't work in certain cancers, and even in the cancers, it works like melanoma and lung cancer. It doesn't work for the majority. So it's a bit more tracing back and why it doesn't work, getting some mechanistic insights. And as you might imagine, this is a cells arranged in space, so you need spatial technologies, and that's where technologies that have come about in the last 10 years, and especially last 5 years, or maybe 2-3 years, have really accelerated these discovery efforts. So just to set it apart, from genomics has been around for many years, So there's the canonical ones is the bulk sequencing. You can think about it like getting a lot of cells from the tissues, extracting some biological material, running it through an assay. So you can think about it like a gimish of different types of cells. And single cells are basically parsing each cell out, of each cell. And you can think about the components that go into this smoothie, basically. So the spatial is exactly the same analogy, but in this case, you actually know where the arrangement is. It's more like a fruit art. So you know exactly which fruit it is, where is it located, these are barcoded, right? So that's spatial technologies in general. And there are many, variants of this spatial technologies. So one can trace back to histopathology, which is the classic H&E staining that the pathologist sees to stage the disease. So these are whole you get an idea of the morphology of the thing. There is actually an AMP histopathology called spatial proteomics. I'll talk about it in more detail, where it's a similar kind of flavor of the technology that can measure, that can tell what is the type of cell, the functional and the type of cell. Spatial transcriptomics that has got a lot of attention over the years, over the recent past. It's basically the transcriptomic, the gene expression equivalent of that. And that tells you for the gene expression programs, and modern technologies are collecting basically any omic now, multi-omic, like the spatial metabolomic, there's spatial, DNA and RNA on the same cell, that's what right. And that's in the last 2-3 years that's been highlighted in many of these top major methods journals as the technology to watch out for. Right. So of course, as a statistician, like what is the actual data that you're dealing with? Like what is the these are images, right, and maps. So from the pathology imaging, so these are the classic H&E stains that the pathologist sees. So these are used to visualize tissue structures, the morphology of the tumor. But what you can also extract is different kinds of features that you can't see, with the human eye, and digitally extract them. One of these features is the inferred cell types. So you can go back and trace like what are the types of cells. So it's an inference, like it's not measured. So you have to do some prediction here, but you can do that. And especially you can't get you can at least get some course level information, whether a cell is an immune cell, or is it a tumor cell, or is it a stromal cell, which is surrounding the tumor. So, the nice thing about this is scalable. It's basically taking pictures of the tumor. So it's imaging based. So it's very scalable. Everybody gets, every cancer patient gets imaging done. So these get hundreds and thousands of patients to do the analysis and mine it. And often most have actually outcome data, some kind of survival or response to treatment, as well as associated genomics data that you can correlate with. As I said, the multiplex is a similar type of technology. It's called immunofluorescence staining. So essentially it is the same thing you take a tumor, you put it on a slide, you basically paint it with different kinds of antibodies. So you paint it with red, you take a picture, you paint it with green, take a picture. So, so that gives you the types of identity of the cells. So these are different types of cells. So the multiplex is basically multiple types of proteins. So you can simultaneously get multiple types of cells, So now technologies can go up to 30, 40 cell types. So these are proteins per cell. And the different kinds of markers here. And again, it's imaging based, so it's very scalable. So this can be gotten cheaply in quotes, and then most have associated. So these are done as correlative analysis in clinical trials. When you have large phase 2 or phase 3 trials, you collect the tissue, the banking, even cohort level trials. The transcriptomic one is a bit more nuanced in the sense that each of these, it's again the same sort of arrangement of the cells, that you have the cells over the map, you get their spatial locations, but now the features are very high dimensional. So basically it's gene expression. So you can think about from each of these spots, you have a 20,000 dimensional vector coming out, which is the gene expression. So it's that's why it's like high end. There's a lot of cells, thousands of them, or hundreds of thousands of them, possibly a million. And then high P, which is a lot of genes. So this is sequencing based. There's a bit more instrumentation involved in sequencing. So even though it's high throughput, it's deep data, but the biological replicates are pretty, still relatively low. Either single digit or maybe early double digit. That at least what I've seen. Right. So that's the structure of the data. There's a lot of analytical challenges with it. Broadly bucketed into two types. One is, these are coming from machines. So there's the whole pre-processing quality control aspects of this. And then, and that can arise in many different, ways. Of course, there's the design, like when it was collected, when was the patient assay. That's critical to know. Batch effects and those sort of things. But having gotten past that, but even like, the type of instrumentation, because these are images, like a red in one might not be the red in the other image. It might be intense. So there's a lot of normalization and standardization that goes. So it's not a solved problem, but some reasonable solutions exist to get the data to a format which is analyzable. So the matrix that we can load up in our Python. But having gone through that, the main modeling comes in. This is basically, at least I feel, spatial statistics on steroids, because we are looking at, hundreds of thousands or million cells per sample, and then we have many, many samples, And and the other sort of, challenge, but at the same time, it makes actually huge statistical problem in the sense that these are non-conformable spatial domains. What I mean by that is classical statistics or geostatistics, is only one space. You measure a lot of things. It's one map of UK or one map of US, right? But then here you're getting tumors, which are completely different shapes across patients now. So to use a crude analogy, the tumors are like gingers. You pick up one ginger, it's completely different from the other. One might be very spiculated, one might be very circular. So how do you actually conform them to do population level analysis when you're doing cross-patient analysis? So, and it's a little bit more challenging in the sense that people do this in brain imaging a lot, but the brain is a common template. Everything gets mapped. So the northwest corner of the brain means the same across individuals. But tumors, there's no such directionality. It can be any way, So pixel ABC in one tumor does not mean the same thing across the other tumors. So it's completely. And I'll show you a high degree of spatial variability.
That's the question on that. So far, everything you've shown has been two-dimensional. Tumors are clearly not. So are we looking at slices, or do we ever get multiple slices?
So it's all two-dimensional. Right now, we are looking at slices. So these are really tiny slices of a few microns that are cut from these biopsies and put on a tissue slide and assayed. So the 3D aspects is an interesting one. I don't know if technology exists to get the whole 3D image of the tumor and the location of the cells. So one of the data sets I'm dealing with is they basically have serial slices and they're trying to recreate it.
Okay, yeah, that's what I guess you'd have to do.
Yeah. So please stop me if you have any questions as well. So I feel this is a sort of a perfect playground of AI and statistical models. Yes.
And this is largely I'm ignorant of the field, but how does this knowledge of this sort of stuff translate to therapeutics?
Good question.
Slowly.
Good question. No, I'll give you a specific example down the line, but right now this is pure characterization at this point. So, and I'll tell you one context which I'll come to later is I'm working with the data sets where it was collected as part of a trial where lung cancer patients were given immunotherapy. So we know some patients responded, one didn't respond, and we have their baseline biopsies, which went through this technology. So we are trying to characterize two things. One, what was different in the tumor microenvironment that predisposed some folks to be responsive to immunotherapy versus other, right? The other is, can we develop a prediction tool based on this that we can predict response to immunotherapy? That's I feel a little bit more involved and a long-term goal. Is there another question here? Okay, good. All right. So just bear with me. This is my pontification slide, and I'll get past this and get to the specifics here. I know we are a statistical audience, but no. So the three aspects to this, right? What goes in, the analysis, and what goes out, right? So what goes in, I feel that the AI can, or machine learning can automate, because if you get a reasonable solution, this is rinse and repeat analysis. So you can do it again and again, and it's actually really good, like, especially deep learning and stuff, to detect some kind of data quality issues. But what it absolutely cannot is fix a bad experimental design. So a lot of this can be automated, and we have actually used some variation of this in one in spatial proteomics, and it's reasonably works well. So that's where the engine, the core comes. That's where you need proper context-driven, biologically informed. A lot of clinicians and my collaborators I talk to, they come in with specific hypothesis in mind, and converting that to actual models. I think there is very lot of utility in there. And I'll show you more about this, but there's so much variability in this that uncertainty quantification becomes a very key thing. And also, as I said, they have some prior hypothesis, or at least some prior knowledge that I expect to see this. And then that's where you can bring in prior knowledge, as simple as what's your confidence level, low, medium, high, that you can build in. Again, I'm preaching to the crowd here. So sort of gets that sense, and that can, it can augment it, but it can't, replace it. And that's where, doing it very organically, is a sort of a sensible approach. But then you go to the end product, like once you've done all the modeling, and that's where, these things can be really accelerated, is like getting really pretty pictures, like some of the students that, my PhD students, they show me. And then the writing aspects of it, but something actually is scientific interpretation. So sometimes even if the collaborator has some ideas, what should be coming out, but actually running it in the literature, like assimilating all the existing knowledge and plausible with plausible biological hypothesis. Actually, it does actually, but you have to pass through a lot to get there. And of course, like shiny app development and coding and all that stuff. So I think that's where it can really accelerate things. If you get the pizza analogy here, the Domino's one, if Domino's is headquarters in Ann Arbor, Michigan, that's where I'm from. All right, so getting past that, so what are some of the canonical and analytical tasks? And there are many. I don't want you to read this slide, but I'll just make this key point where, you get this data from imaging based or sequencing based. A lot of this pre-processing, as I said, can be automated. A lot of these analytical tasks are exact analogs of what is done for standard multivariate data, two-group testing, clustering, and in spatial, there's region detection, which is basically like what we call disease mapping, because there are a lot of disease mapping, and it's basically some region detection, like what are the hotspots in the tumor. And it's just that the data is now a little bit of a different flavor. And a lot of methods have come about, especially from the computational biology realm, which actually uses process-based modeling, but almost most of them here are based on heuristic metrics. It's using variations of statistical. So I'll give you a few examples. I won't go into the weeds too much into this, but at least give you an idea about the type of models. So one from histopathology imaging, And it's a very simple problem of quantifying cell-cell interactions. And then similar concepts for multiplex imaging. And if I had time, I don't know if I'll have time to go into some, recent work with network models for spatial data. You about in network models here, so at least give you the high-level idea. I'm happy to chat later about specifics. So, no. So the cell-cell interactions. So this is a specific data set we worked in melanoma. So melanoma is known to be a very highly immunogenic cancer. It's called an immune hot cancer. So there are immune hot cancers and the immune cold cancers. So melanoma, lung are immune hot, breast, bladder. We expect a lot of immune activity. That's why it responds by and large well to immunotherapy. So the idea here is, so this is more retrospective analysis. So we have these H&E slides. So a collaborator of mine worked, we worked on a machine learning based model to get like the cell locations and classify them as basically two types of dots, red cells and black cells, right? The red cells are immune cells, black are tumor cells. And then a pathologist, first trained, told us which cell, and then we trained a model based on it. It's a random forest based model. And then we went iteratively a couple of times to clean up the predictions. So anyway, so at the end of the day, what we have is the spatial map and the identity of the cells. So, and as I said, that's 100,000 cells per biopsy. We have around 330 biopsies, and also this comes with associated multi-omic data. And it's actually a subset of the TCGA data, the TCIA data. So this is the additional processing that we did in-house. So when my student and I actually started looking at this data, many things stood out, but more importantly was the amount of variability that's in there. So this is just the two biopsies here, and like looking at a very small portion of the biopsy here, and so you can think about two types of variability. It's just one is the first order, it's the prevalence of the cells. Right, at different regions, there's different prevalence. That's the first order, and the second is the interactions relative to each other. So, so the first task was quantifying and understanding this variability. And as I said, this might have implications for immunotherapy. So the first question we sort of very simply put is: can we classify within these biopsies? At a reason, like, can we come up with a metric, like a probabilistic method to spatial interactions? So think about just simply three types. There's a low negative interaction, which means they're separated out, by some distance. A null interaction, they're adjacent to each other, and a positive interaction, which is like they're mixed with each other, right? And to give you a scale of the idea, we are talking microns here, right? To give you an idea, a cell is 5 to 10 microns. So the sphere of influence of a cell is actually 15 to 30 microns. It's very localized interactions that we're trying to assess.
The spatial aspect of this, if you sample the same tumor twice, how much does the process of taking the sample out, chopping it up, distort the spatial aspect?
So in the H&E, I think you can get high fidelity because these are whole slide images, right? These are whole slide images. So this is you get whatever part of the tumor you biopsy and you get that. And this slide is actually what the pathologist sees to do the staging. Yeah. So you hope that they get it right.
I'm just wondering if you're training these things to recognize facial structure. If I applied a slight shearing force to my sample, would it be sensitive to that? And maybe you're augmenting the data such that those sort of things are averaged out, or?
So there's a couple more steps that's going through this. The first step we do is like, these are pixels, right? So the first step is actually to segment out the cell. So you need to know the location of the cell. So that can be done. You segment out the cell, then you map the pixels to the cell, and then you call the cell either a tumor cell or an immune cell. So these are not done at a pixel level. So is there any uncertainty in that? Of course, there is some uncertainty in that. And that's why we had to get a pathologist to say, you see the cell, is it a tumor cell or a stromal cell or immune cell?
Good.
Right.
So I imagine the pathologist is working more on topological views of the data, right? They implicitly know, they have some mental model of what the
Correct.
cell
Yeah, how distorted the cell is, the morphology of the cell gives a lot of ideas. I mean, for them to say, is it a tumor cell or not? And the intensity of the markers also tells you, is it a tumor cell or a stromal cells? So to a reasonable degree, you can do it because it's measuring what it's supposed to measure. So if you believe that, you can do it reasonably accurately. Yeah, because this is the starting point, right? If this breaks down, everything else is rubbish, basically. And I'll tell you, like when I come to the spatial multiplex imaging part of it, it's a little bit more nuanced there. No, calling that, and that's where we have to do some uncertainty quantification. Yeah, but for H&E pathology imaging, it's pretty. So no, I mean, so yeah, so just going back, so we are trying to quantify interactions this way, and this is not a new problem. Ecologists have thought about this a lot, like geology. It's a spatial mark point process. What is a mark point process? It's a random point cloud. And the marked comes from the fact that each of these spots have they're either a red or a black, depending if you are immune. And then in this case, it's a binary, but it can be multi-category also. You can have 3 types of cells, 4 types of cells. That will be in the multiplex imaging when I come to it. So essentially, you can treat each image as a realization of this process. So there are many marked point process models. There is one in particular, actually, which is very natural for this data, which is called the hierarchical Strauss model. And it's a very cool, nice model, which is linked to you can link it to the actual tumor evolution and how the data could have potentially evolved. And I'll tell you the reason why. So without going too much into details, this is the likelihood function. But one nice thing is, it's based on accounting Poisson processes. So there are 2 inhomogeneous Poisson processes that are linked by this parameter gamma, right? So that's the key takeaway here, that this captures the interaction between the interaction that I just mentioned in the previous slide. You want to infer this parameter basically, based on this likelihood. So we didn't invent this. This has been around for 50 years and it's used a lot in ecology. It's very natural for the tumor cell data for a couple of reasons. One is this hierarchical means that it's conditional on one, the other came. So what it means is basically, in the tumor evolution process, cancer happened, right? Tumor cells formed a collection of tumor cells, and that's how the tumor mass evolved. But then, as I mentioned before, it did a systematic recruitment of these immune cells, right? And then they came. So the similar underlying thing is in ecology, where say a tree grew first and then the weeds grew around it. And how did that come about, right? So that's the hierarchy here. So it's conditional on that. And the good thing with this is there are many point process models, but this one actually allows both positive and negative. So the bidirectionality that there can be a repulsion and attraction together. So the nice thing is it's a likelihood, or it's a generative model for the data. Good or bad, that's a different thing, but at least it's a starting point. The fitting is actually, really easy. It's complicated in the sense if you work with it directly with the likelihood, but actually you can as an approximation, a reasonably good approximation that can be done, which actually you can fit it using a GLM, basically. You can run a standard GLM, and that's called the Poisson trick. Basically you're estimating a regression parameter. So this turns out to be a regression parameter. We have 3 regression parameters. One is the prevalence of cell type 1, prevalence of the other cell type, and the third one, it gives you the interaction. It's that simple. So once you get it down to the regression, you can put a lot of priors and learn that. And so that's what we do. We fit the Bayesian way, and then you get a posterior distribution of this. But it actually turns out it's a little bit more. Oops. Better this way. So actually, I'll make the point here. So what happens is you get a value, right? You get a value of this, like you get a 0.1 or a 0.2, and you get an associated distribution. The problem then becomes it's an unbounded parameter. So how do you determine significance? How high should this value be away from 0 to determine significance? Should it be 0.2? Should it be 3? Should it be 30? Should it be 100? So there is an extra level of inference that one needs to do on top of it. And it actually became a sort of acute inferential problem of how to determine significance of gamma, basically. So that's where we had to look at it, think about it a little bit more closely. So we came up with this, I'll tell you the gist of it, with this inference measure, which measures puts it on a probability scale. So take this parameter gamma, which is on the positive real line, and computes how far is it away from a null distribution, right? And the key point here is this null distribution changes with the data, because the null for one biopsy might not be the same for the other biopsy. And so essentially, it's basically looking at the intersection of two densities, this probability. One is the posterior and one is the empirical null. So it makes it nice that this cell type interaction probability, what it is, the intersection, you can compute it from MCMC samples, and once you have the null, and then you can compute this within cross biopsies. So I'll tell you just the idea of it. So if you think about a particular biopsy, you strip out all the immune cells, you take all the tumor cells, because those change across biopsies. And then you generate, n null distributions from this, under a homogeneous Poisson process. Then you compute the posterior distribution of that, and then you get the empirical null, and then you have your observed posterior, you just see the intersection. So it's a very simple idea. So the intersection tells you how far away it is. Captures the point being it captures the uncertainty, but at the same time you can determine, on a probability scale, how, closer to 1 is higher tumor immune infiltration versus not. Did you have a question?
Yeah, I have a question. When you talk about null distribution, is it the picture that you showed us? Is that what you're assuming, that in terms of the interaction, or is it something different?
No, so this picture is what I wanted to capture, right? You meant this one, right?
Yeah.
Yeah, so I want my gamma to be a high value here, right? I want it to be high positive, high negative, and close to 0 here.
Okay.
Yeah, but in reality, I'll never observe such cleans. So I might get a value here of say 0.1, right? I might well get a value of minus 0.2. Right, but I want to know, like, relatively, it's hard to calibrate how high is high. That's the. So putting it on a probability scale makes it a little easier. So closer to 1, because and then I'm taking the posterior uncertainty into account. That can change. The point being that can change across biopsies. Yeah. So putting the whole thing together, it's So as I said, these are very localized interactions. So the sphere of influence is very localized. So you can run it on the whole biopsy, but we actually partition the biopsy into multiple tiles, and then compute it across, and then basically come up with these maps. So, so this is the probability of interaction with this tumor immune. And those are the darker it is. You can see around the edges, there is a lot of interaction going on in some cases. But the lighter ones, there's less. So, and once you have this number, you can average it, to get sort of an overall number. So we did this in the entire biopsies, and you can see this is the scale from 0 to 1, the average cell type interaction probability, and you can see it's on the right end. So it's a little bit of like a positive sanity check, because you expect melanoma is a immuno, so we expect higher immune infiltration. There is some variability across the patients, but overall it's pretty high, reasonably high. And some patients don't. Then we use this actually right here, yeah. So we use this to see, does it have any prognostic benefit? So we have overall survival on these patients, and adjusting for the usual cancer stage and the clinical and the demographic features. Does it have any sort of associative ability above and beyond? So it's not a lot, but it does have some it's slightly away from zero. So, but still, it has something that it's some information. We also actually did an association of the tumor immune infiltration with omics data to figure out if there is something molecularly associated with it, like gene expression signatures, like DNA mutations and other things. So just to characterize them more going into the biology.
So just a question on that. Wait a minute. So what you're saying here is the more infiltration you've got, the higher the hazard?
Yeah.
I might have thought that would be the other way around.
That's a good question. So that puzzled us as well. Now it depends what you call immune cells here, right?
Ah, okay, right.
Now, I'm putting all immune cells together. And that's why we had to look at the molecular data, is what kind of more immune cells it is. It turns out it's actually more neutrophils and macrophages.
Right.
So that is sort of bad.
So you don't have kill, T cells or
Exactly.
or the right kind of T cells.
Yeah.
Okay.
So, yeah. So when Nate, the student, actually, I should give credit here. Nate was the one who developed it. When he first showed it to me, this doesn't make sense. So that's why we had to go back and ask our collaborator too. That's, no. All right. So much time to okay. Few more minutes. So one thing, if you caught on here, right? Like one thing is we average out this thing to come up with a. So that sort of bugged me and Nate. Like, can we, we are ignoring a high level of spatial variability within the tumors here. So, so the second step was, can we look at this, spatial variability within the, and naturally, what the question you asked, John, like, why did it confer increasing hazard here? So one thing we try to is integrate with gene expression data to explain the tumor microenvironment. So the basic question was, what was in the molecular architecture of the tumor, like as assessed by gene expression, that was associated with the tumor immune infiltration. So essentially you can think about this as regressing the pathology images on the gene expression. So, so that's the regression problem here. So as I mentioned, these are non-conformable spaces, So, so yeah. So this is essentially what we try to do. So we have this the original image, we have this CT bio, this cell type interaction probabilities or the interaction parameter, and on each of them we have a gene expression vector. What is explaining this? Right? So open it again. Sorry. Did it in Google Slides, and usually the equations come out. So but I do have yeah, maybe I'll work with this. Yeah, share the slide. So maybe I'll explain this here. No. So essentially, the idea here is you have these different biopsies, and so these are my outcomes. And then each of them, we regress it on the gene expression matrix. So those are the covariates. Okay, so that's the general problem.
So the gene expression, you've got a single sample for each tumor or
Correct. So it's at a patient level. So it's an N by Q vector.
So you don't have a single cell for each.
We don't have single cell. This is not single cell. Yeah, so this is bulk gene expression. So that's where a little bit more careful model building has to come in, where you have a population level effect, right? And that population level effect is actually what is the main effect, and that basically drives the between patient heterogeneity in this. So that's at the population level, and this is actually a random effect, which is a spatial random effect, right? And you can see that the space is changing with the patient, right? Because each, so this is a little bit of a different regression model than usual spatial regression models, that the space changes, and so you have different degrees of smoothness within the patients. So that's controlled by a car prior, so that I won't get into the mechanics of that didn't show up. But the essential idea is, that each of them has their own spatial process, and then you can do a selection of this, because we're still doing population level analysis here across biopsies. So those tells you the genes that might be driving the within biopsy, because it's a random effect. It's a spatial random effect. And then it's actually a covariate dependent spatial random effect. So, and then you can determine genes which are driving this. And as it turns out, actually, the top ones that come about is actually the neutrophil class of genes. And that's what was explaining most of the tumor immune infiltration, and hence positive effects on hazard. That's what we found out. So through modeling also, but pure simple exploratory analysis also threw up that. But then we looked at a larger panel of genes here. So some were consistent, like this. This neutrophil class wasn't actually there in the literature that we found, but that was something normal, but there were some usual suspects that already popped up in this.
Sorry, I have one question. How did you go from the genes to the cell types? Did you use something like the convolution method, like CyberSort, to classify, to extract the proportion of each?
Oh, good question. Yeah. So we didn't use all the genes. So the genes belong to particular immune classes. So we only subsetted to the genes that belong to these cell types. Yeah. Yes, that's right. Yeah, we didn't do deconvolution here. The deconvolution, data we use to explain which type of immune subset might be overrepresented in the data. But here we use the actual gene expression. So the neutrophil class might have 20 genes. This might have 30. Yeah. So, yeah. So, I'll admit this is a little bit looking under the lamppost a little bit, but at the same time, looking at immune signatures, it gives a little bit more. You could run this across the whole 20,000 genes, but it's more computational rather than. Right. So I have a few minutes. I'll just tell you briefly about spatial multiplex imaging, because this is something actually sort of caught my fancy, and a lot of work going around this, a lot of data sets that are being generated, both internally and externally. So, so this is, as I said, it's the same flavor of data, but instead of H&E images, these are now spatial proteins. And then you still get raw images, but it goes through what is called a cell phenotyping. So what essentially it means, as I was saying before, right? You have these images, you have to segment out the cell, you have the pixels, and then from each of these cells, you get like a vector of proteins, say 10 proteins, right? But then, they have been measured for specific cells, like each protein maps to a certain type of cell, but it's a continuous expression. So there is something called a cell phenotyping, where you basically draw a histogram across cells and do some histogram thresholding to see if it's a cell type or not, right? Is it positive for, say, is it a T cell or is it not a T cell? Is it a B cell? So there's T cell markers, B cell markers, and so on. No. So this is a little bit of, I feel, an open problem and something that, AI can automate. We are trying to do that, but it's still an open problem because, these are continuous markers and we are trying to discretize them to call them different cell types, But for certain markers, it's easy to do, but for certain markers, it's not very easy because, you don't get this zero inflation most of the time. So we don't have an optimal solution. You can maybe work with the continuous data directly, and maybe that's a better strategy. But for interpretational purposes, going to the cell types makes it a little easier. So again, you get like, instead of 2 cell types, you get multiple cell types. And then again, the characterization is pretty similar. You have the first order, which is just the prevalence. The second order is the interactions and the quantification. Then you do like association with outcomes and all, right? And that's where we need statistics. All right, so I already said this. So there are a lot of modeling issues with this also, and that's where I said, I'm preaching to the crowd again here, you need careful statistical modeling. And the reason is this, twofold, at least two. One is there are rare cell types, that means not all cells will be present in the tumor. The other is actually a lot of technical artifacts sort of come in, which is like holes and inhomogeneity. There's a lot of spatial inhomogeneity here. And for example, this slide right here, you see, this is actually, nothing has been assessed here. The tumor actually tore, so there was a shear in the tumor, right? So the raw data for this is just taking a picture of the glass, the tissue glass, right? So when you map it, you actually, the data is not observed there in these, so you don't observe any data. So you can't put through a vanilla AI system and try to solve that, because you'll get some background intensity and it might map to something. So the essential point being that because of this inhomogeneity, you need some sort of a robust and scalable metric to quantify cellular interaction. So the basic question is still the same, like, what is the interaction between different cell types, 2 types of cells, right? So that's why we've developed this pipeline called Dimple, but I'll give you the key idea here now. So it's still a point pattern, but now the point pattern is multi-type, right? You're showing 3 types here, just for illustration, cytotoxic T, PD-L1, and T helper. So we do actually very simple things. So we take this whole thing and estimate the spatial intensity, the intensity of this point pattern. You can estimate it using a model-based way, or you can do something very simple, which you can do a 2-dimensional current density smoother, spatial smoother. Okay, so you get like 3 of these, right? You get this for all the PD-L1, which is these blue dots. You can get it for cytotoxic, which is all yellow dots and this. And all it needs now is how far are these distributions away from each other? So that captures the interaction. So if they are right on top of each other, the hills and valley matches up for this, right? You expect them to be co-localized. If they're far away, you don't. And there are many metrics for computing distances between densities, either geometry based or, information entropy based. So one is Jensen-Shannon distance. And as you can see, like these two, it's pretty high. That means they're far away from each other. So the higher distance means they're far away. And 0.3 here means they're closer to each other. It turns out actually this works remarkably well. And I'll show you one example here, right? So essentially, the essential idea is once you get the distributions, you can compute this cheaply for each image because you're using the entire spatial map, right? So, and it's bounded. So there are some distances which are not bounded, so it's harder, but it's 0 is perfect overlap, 1 is no overlap. So I'll show you an example here. So this is the actual data. This is actual data. So these are CK, which are tumor cells, and these are CD19 cells. So you see that it's perfectly non-overlapping, right? Like, it's like a donut shape. So if you just drag here, if you go CK here and CD19, it's yellow. It's closer to 0.8. There's no overlap. So if you take these two, CD14 and CD19, CD14 and CD19, it's on the lower end, right? So that means, there is there's no overlap. Oh, sorry, there is overlap. It's closer to 1. Oh, wait, closer to 0. That means there is overlap. I keep getting these. And we have done a lot of simulations for this too, like a lot of inhomogeneity with the tissue, like tumors with holes and different types of patterns. And it's actually turns out to be pretty robust to that, because mapping this to densities and then measuring this, there is some level of robustness that comes to it. And once you get this, think about this. So essentially what we are doing is we have lots of patient level data. Oh, I forgot to mention one more thing. There's a lot of subsampling within tumors here. I think it addresses partly your question. So these are collected at different spots along the tumors. So you get like 5 images per tumor or 10 images per tumor. Then, there's repeated sampling here. So it reduces the whole image, this whole image into a one number. And now you can use it for any kind of downstream modeling. Maybe you can run in a clustering model or a prediction model, any kind of model too. And then you can build a random effect.
And if you have several images, do you take the average because you compute this per image?
Correct. So it depends on how you put it in the model, but you can use a random effect model to get like intra-patient variability, inter to intra-patient variability.
You look at the intra-tumor variability.
You've got multiple pairwise comparisons here. So what do you actually put in your downstream model?
So that depends what you're looking for, right? So if you have, say, I have an example here. So when we are looking, you can do univariate or you can do multivariate. So here's a simple univariate, and this is the response to immunotherapy that I was talking about. So we just ran a simple univariate test. So we have responders now under 2-group testing. And the ones that you see are actually the ones that are significant, adjusting for multiple comparisons. Okay. Or you can take this whole vector and build a prediction model and see which ones of these is predicted. The one thing is you have to be a little careful in doing prediction here because this is inherently tied with the prevalence of the cell types. So you have to adjust for that. Like if you have higher prevalence of one cell, intuitively you'll get more interactions. So you have to adjust for that. Then actually you can extend it to 3-way interactions. And this actually came while I was talking to a collaborator. You have 2 cells, but it might happen that you have type A and T cells and B cells. But if you have macrophages, a type C, that might end. Can you do 3-way? No. And actually you can do 3-way because we're estimating densities, right? So if I have my 3rd cell type, I can get at different quantiles of that densities, I can get different interactions. So I can look at parts of the tumor where the interactions might be different. It gets a little complicated interpretationally, but at least it can be extended if needed, But again, you have to know where to look, basically, for 3-way interactions. So we actually run this for many data sets, in-house data sets. And so one is response to immunotherapy in lung cancer, and it turns out like, this PD-L1 negative APCs is an important one, the interactions with T regulatory cells. So I don't know how many of you know, like T regulatory cells won the Nobel Prize this year with discovery of T regulatory cells. So this is another sort of MHCI status in lung cancer. The tumor immune interactions were significant. So it's just not for response, but other mutation status. So this is actually a project where they wanted to use IHC to predict mutation. So the idea here is mutation genetic testing is, it's involved, and then and then there is some level of sequencing. So these data are cheap. It's immunohistochemistry. So can we develop a prediction model that can, basically replace genetic sequencing? at least ARID1 mutation, which is in gastric cancers, it's a major mutation. And it's a similar principles in immunotherapy. So this is my collaborator, Tim Frankel, who actually wants, he thinks like this is nice, rich data that you can collect in resource-rich settings like UK and US. But we want to drill down because maybe you don't need that many markers. What is the minimum set of markers if we had to scale this and deploy in a resource, less resource places? And that's the prediction one I was mentioning. And so we're just starting on that, like trying to use spatial biomarkers to predict response to immunotherapy. We have only one data set, 200 patients. We have to do an external validation first of all, and then maybe take it down the line. So that's a little bit of an aspirational line dream right now to develop a assay that can be deployed more widely. But I also believe there is some sparse subset of these spatial biomarkers that can predict immunotherapy. But which ones are those? That's where we are trying to back right now. See, I'm over time, so I'll skip this one. This one, but just and so a lot of this actually for the last one, the spatial multiplex one, there's actually we have a GitHub website. All of these have softwares freely available. But this one actually has a shiny app where the lung data is already provided, the previous lung data, and then actually anybody can upload their images and run the analysis. So it's running on a POSIX server, and it can run the distance space because it's very simple. It's just 2D kernel smoothing and computing Shannon-Jensen. So, it can be done in parallel. So let's see. Yeah, now. It's very, if I haven't already, shared my excitement, it's a very exciting field. That's something that I've pivoted into. Lots of open problems, especially for spatial analysis. And it's a good, perfect playground for AI models and AI and statistics. Yeah, and the key is, this is still on the hype curve of spatial technologies. It has to land to make an actual impact, like clinical translation. And I think somebody asked me here, right? Where will it finally land up? And that's what we are heading towards, at least with the imaging technologies. I can't speak the same of spatial transcriptomics. For a discovery, it's a fantastic tool. But clinical prediction, that's what it is. And then, yeah, so 2D to 3D, that's very interesting, actually, there's a lot of efforts on mapping the whole atlas of cells in tumors and combining with multi-omic now that. So I'll end there. Hopefully, I've happy to take any additional questions.